Hello, everybody. So I'm Professor Alawalia, Amrita Alawalia. I'm the Secretary General for WCP 2023 and also one of your chairs. And I'm here with Dr. Natasha Karp, who's my co-chair. I'm from Queen Mary University of London. Natasha?
I'm from AstraZeneca as a Director of Statistics.
And we have a fantastic session for you today, but actually Natasha and I, together with our colleague Ben Phillips here, are hijacking the first 5 minutes of this session, and I will let Natasha explain why.
Okay, so we are actually running an experiment here at the Congress. We are testing the effect of an intervention to see if we can change people's beliefs and attitudes around sex-inclusive research. There are 3 test groups. There's a general population, there is a population who's interested in the topic, and there's a population who attend the intervention. You are our control population. So we need your data. It's a survey with around 24 questions where you press the button and it just answers your questions. The only data we keep on you after telling us how many years you've been in the industry and your age, but the only identifiable data we keep from you is optional. It is your email address. If you give us your email address, you'll be put into a draw with the chance to win one of 10 £50 vouchers for Amazon. Or if you're abroad, we'll work out how that will work when we get there. But we're hoping to use this data to show that the intervention works to support the conversion of the material that we developed onto an online resource and help pass it out globally around the world. So if you scan this QR code, but if you do have any problems with that approach, just reach out to one of the support staff. He's got his hand up there, and Ben as well at the front here. He'll come and help you with the process. Ideally you need to be an active researcher working with animal data or ex vivo. So you're using animal samples, and then you'll be asked a series of questions about what do you think and why do you do what you think you do? You know, what's your challenges?
So we're going to just give you 5 minutes of silence to complete that survey for us, please, before we start talking to you. I hope that's okay. So the first speaker in our session today is my co-chair, Dr. Natasha Karp. And Natasha is going to kick off the session before we start focusing on sex differences in cardiovascular disease, on why designing the right experiment to assess sex is important. Natasha.
We'll actually have a different title, but let's go figure.
Okay.
Hi, thank you very much for coming to this event. It's so great to see people interested in this topic. I'm going to be talking about one size fits all. What's the current thinking about sex-inclusive research? And as I said, we're actually running an event here at this event to try and understand people's perspectives and whether we can alter and address some of the misconceptions and the barriers that I'm going to talk about with an intervention. And you are our control population, a group of people who are interested in the topic, so therefore will be more comparative to our intervention group. So as a disclosure, I actually did sit on the Medical Research Council group in the UK, talking about experimental design that's recently led to the recent mandate moving towards sex-inclusive research. I sit within AstraZeneca as a Director of Statistics, leading a team of statisticians supporting preclinical research, and I also collaborate and work with the NC3Rs on their various working groups supporting animal research. So as an outline, I'm going to touch upon terminology, whether we think sex matters clinically, what is actually happening in the preclinical arena, what is sex-inclusive research, what are the barriers, and some conclusions. So, terminology. Sex, gender, does it matter? People often find the word sex a little bit of abrasive and uncomfortable and tend to use the word gender, but they actually have different definitions. Sex refers to the biological and physiological characteristics of males and females, where gender is related to the social constructed characteristics of women and men and relates to behaviors and expectations and values. Gender is as a social construct will vary around the world and actually will vary with time. Both sex and gender influence health outcomes. For example, gender influences people's access and ability to engage with healthcare or their choices to engage with healthcare. So when we are conducting preclinical research using in vivo or ex vivo or in vitro, we are actually considering the sex, not the gender, and therefore we're now encouraged to use the terminology sex to avoid the misrepresentation of our research. So clinically, does sex matter? Well, this is one slide on the topic. I know, or I hope you guys have seen from your personal experience, that a prevalence of a disease, the symptoms you're going to experience, the progression of a disease, the side effects, all depends on one's gender and sex. And if we take the topical case of COVID-19, there's a lot of papers on this, but if you just quickly look around, there's a general consensus that the prevalence of COVID was higher in females, but there was higher morbidity and mortality in males. If you look at this from a sex difference, there was a higher expression of the ACE2 receptor for the virus in males, and there are immunological differences driving the different reaction. If you look about gender, we can see that women tended to have more social contacts and work in care roles, increasing their exposure, but that men had higher rates of smoking and drinking, increasing their risk of mortality and morbidity issues, and had a lower uptake of preventative measures, increasing the dose of the virus they received. So you can see that both sex and gender are playing out in a disease outcome. So what's happening in the preclinical research space? Well, preclinical research is experimental research. So we simplify a complex world into a testing inference space where we can isolate cause and effect. We have a control group and a treated group, and the only cause of the difference can be the intervention. So we have to simplify to make the space doable so we can answer questions incrementally. In vivo research, we tend to take our pool of animals that are of one sex, one genetic background, one age, one diet. We test the intervention, measure the outcome, analyze the data, and then we generalize. This is also a simplification, which is part of the experimental process, but it's also driven by the need to decrease the N because of our ethical implications of using animals. This strategy has led to an embedded neglect of sex within preclinical research. And this is just some of the papers talking about the issue. And they are spanning the whole spectrum of research. So we see it in the reporting, in the design, and in the analysis. Frequently, the sex is not reported in manuscripts, as though it's not a relevant feature. If you look at experimental design, Beery in 2020 looked across 9 fields of biology, and she did the study back in 2009 and again in 2019. She had found that the proportion of papers studying both sexes had increased in 6 out of the 9 fields of biology, but the average was 26 to 48% of studies included both sexes. And in fact, pharmacology, unfortunately, had a reduction to only 30% of papers included both sexes. And we likewise see in the in vitro space that we typically only study one sex of cells, and actually the majority of cell lines are from a male source. When we analyze our data, if we happen to breed our animals in-house, we get both sexes, but what happens then is we tend to pool the data together as though sex isn't a relevant source of variation. And recently, there's been an uptick in the number of papers reporting sexual difference, sexual dimorphic effects because of the NIH mandate. But though when they are doing so, 1/3 did not test statistically and back up their claims with a statistical test. So we have 2 problems playing out here. We have inclusion of both sexes and then the appropriate analysis, visualization of the data. And there is now an emerging evidence that our knowledge base is biased. And this slide is based on material by Professor Mogul, who works in pain research. And he had written a paper exploring that there had been an increase in the number of papers studying both sexes since the NIH mandate. And there had been an increase in the number of papers identifying sexually dimorphic effects. When he went and looked at those sexually dimorphic effects, that was because 72% of the effects were only seen in the males, and 27 were only seen in the females. And he argues this suggests that our underlying knowledge base from the historic position of only studying males has led to a knowledge base that is biased towards male biology. And the diagram on the right is from one of his slides that he's kindly shared with me, and it's showing the signaling pathway for pain, and he is arguing that a whole branch of this, and he's got numerous papers to back up his argument, is actually specific to males. So even though the phenotype outcome is the same, which is pain, actually the underlying biology is different between the two sexes. And the reason I got into the topic as sex-inclusive research was due to my earlier career in academia. I was working on the International Mouse Phenotyping Consortium, which was a large project to relate the genotype to the phenotype. So what we were doing was knocking out every single protein coding gene of a mouse and characterizing the mouse through a standardized pipeline. And if we look at the scale of the project, there were 10 institutes. I had data from 14,000 wild-type mice, 40,000 mutant mice, 2,000 different knockout genes, and I was looking up to 234 traits. When I joined the project, they were originally separating the data by sex and analyzing it. And what I did is I came in as a statistician to build a pipeline to ask, what is the effect of the knockout and does it depend on sex, after accounting for a sex difference? Now, this data set gives us the ability to ask how often does sex just generally play a role? Because there's so many different knockout events, it's like a general research question of how often this occurs. And this was published in 2017. And what we found is that the first question you can ask is how often does sex change the baseline data, just in the control data set? So if you pooled the data, this would add to variance in your data. So we can split the variables into two types. Categoricals would be things like rib shape is normal or abnormal. Continuous could be things like sodium level, lean mass, etc. And what we found is the categorical variables, even though they're rare events, the abnormality rate was different in the wild type between the males and females. Likewise, in the continuous variables, there was 60% of the variables here had a baseline difference between males and females, and that is a common status. But maybe what's more interesting to ask is how often does sex modify the treatment effect? So in this case, the genotype event. So here we're looking at pie graphs where the genotype was shown to have a significant effect, and then I asked, does it depend on sex? And we find for the categorical variables that had a significant effect, 13% of the time it did, and for the continuous variables, it was around 17% of the time. Now, these experiments weren't powered to detect sexual dimorphic effects. We had only 7 male and 7 female knockout mice per knockout line, but the proportion of sexually dimorphic events is significant, which suggests that sexually dimorphic effects are common. Okay, so if we know from our own experiences, and we know from the research, and we know that there's been a sex bias reported for over 25 years, why has nothing changed? So I'm going to switch to some exploration of the barriers. So this is a sociological exploration where a person has interviewed scientists from the US following the NIH mandate. And they identified 3 different themes. They identified that scientists believe that generalizability is important and sex matters. It is important to understand biological differences. But science needs us to avoid complexity. To make progress in science, we have to make the problem doable, the research question. And so practicality, they didn't feel that they could study sex in a doable way. The majority of issues they were talking about was cost. The MRC ran a survey prior to the formation of the working group to understand the issues from that community. And the majority of the researchers, whether they were in vivo or in vitro, saw the benefits of considering diversity and translatability, reproducibility, and the ability to detect sexual dimorphic effects. But they were worried about the costs, and because they were worried that it would increase the number of animals used, which would also be against the ethical principles under which we operate our experiments. So let us step to what actually is sex-inclusive research. Well, this concept of a sex-inclusive research has really been driven because funding bodies are driving change. They're moving from a position of recommendations to sort of mandating that inclusion will be the default. And the latest in the UK is the MRC announced last year. And I understand, talking to some people here at the event, that in Australia there's also a bit of a push. So it seems to be a very global, worldwide phenomenon. The SAGA guidelines are quite a sort of useful back point. So they say that the first thing is we need to use the term sex and gender carefully. But also what they're saying is when you need to consider sex and gender is when you can identify the sex or the gender of your subject. So if we can use chromosome testing, then we know the sex, so therefore that is the point at which we should start considering sex-related differences, even if these were not initially expected. They also give some reporting advice guidelines. So for example, if you only study one sex, they wanted up there in the abstract that this data only comes from one sex, and this limits the potential generalizability and a discussion of whether you think sex matters. They want the data analyzed to see whether sex has an impact and disaggregated by and summarized by sex. Now, these reporting guidelines have now been taken on board by a number of journals. And so, for example, this was a paper published in 2022 by the Nature group that said they are raising the bar on expectation. If you don't follow the SAGA guidelines, your paper will not go out to review. And basically, they've implemented a workflow to assess that you're aligned with those expectations. So what is the expectation? Inclusion is the default. So historically, to include both sexes, you would have had to justify that it was really important to include both. Whereas now, you have to justify the exclusion. So first of all, the requirement is to specify the sex. The second is to include, and the third is that your analysis should account for sex as a source of biological variation. But let's explore the vision. The vision isn't driving you to identify sexually dimorphic effects. That's not the goal of the inclusion. The goal of the inclusion is to estimate generalizable results, an average effect of the two sexes. And if the effect is significantly different between the two sexes, then this will become apparent and suggest that there's maybe something that's biologically very important that you should then start following up. So you're actually powering your experiment not to detect a sex difference, but to detect the treatment effect across the two sexes. What are the exceptions? When you cannot determine the sex of your sample, if you're studying pure molecular studies, if you are working with sex-specific conditions or phenomena, e.g. if you're working on ovarian cancer, if you are acutely scarce resources, and if you can provide strong justification. Now, that strong justification was pulled out by the MRC that female variability is not sufficient justification, and that absence of evidence to date of sex differences is not justification. And actually, back in 2008, I had written a paper exploring the barriers and the misconceptions, because I would argue, when I talk to scientists about sex-inclusive research, the initial resistance is not because they believe sex doesn't matter, but because they have a series of misconceptions, a skill gap, practical concerns, and concerns around the 3R interpretation. So the most common misconception we're seeing is that females are more variable. And this is why the research is predominantly focused on males, not because people are more interested in males, but they believe that if they work with male animals, the variance would be lower, and therefore you would use less animals. Now, this concept, there's been a number of pieces of research, and I'm pulling out two, one focused on rats and one on mice, which are meta-analysis where they have collected both sexes, comparing the variance between the two sexes, and found no evidence that the variability is inherently higher in the females compared to the males. So whilst it might be an embedded belief, empirically the data is not supporting that belief. I would also like to argue that inclusion isn't at odds with the 3R mindset. And there's two slides on this, so travel with me here. The first of all is when you're breeding your animals, you're actually producing both males and females. And if we only take the male animals, the females are being discarded. But it's the N beyond your experiment. But actually, it is more efficient to include both sexes in your experiments, and I'll show you that now in the next few slides. But actually, if we look at the definition of reduction, it has changed. There is a standard definition. We are focused on the number of animals used per experiment. But there is now a more contemporary definition that has been around for a lot longer, which is say that experiments should be robust, reproducible, and truly add to the knowledge base. But I do think as a community, we're very much focused on, I've used 5 animals per group or 6 animals per group, rather thinking more holistically about our animal use across our experiments. There is a misconception it will double my animal use. And I've heard it from right up to professor levels of people who are involved in reproducible research. If I study both sexes, I'm going to double the number of animals I use. The actual advice is to keep doing what you're already doing, but change half the animals in your study to female. Now, this might feel counterintuitive, but I'll explain that shortly. And there are 2 papers I'm referencing as evidence, and one of them is Ben Phillips here and myself's paper, which uses simulations to explore how, when we add signal in and variability, to show and to demonstrate that including both sexes does not decrease your power. And that's because you're moving from a complete randomized design to a factorial design. So on a complete randomized design shown on the left, you have a pool of animals and they're randomly allocated to control or intervention. You might use something like a t-test or a Fisher-exact test to ask, does your intervention have an effect? You're basically saying, what is my treatment effect? And you're comparing that to the unexplained variance. When you switch to a factorial design, you have your first factor of interest, which is your intervention, such as a drug or a knockout. And you have a second factor, which in this case is sex, whether it's male or female. And then when we actually do a factorial analysis, and we're asking for my outcome of interest, what is the effect of treatment after accounting for the baseline sex difference? And you also then ask, does the treatment effect depend on sex? You've accounted for the baseline sex difference, so when you're asking for your treatment effect, you're then benchmarking it against the unexplained variance. So that baseline sex difference doesn't add variability to the data, it's accounted for the model, but you also get the benefit that you're asking this interaction, you're doing a statistical test, does it depend on sex? And this is where the common approach being used, such as pooling or disaggregation, falls over. If you disaggregate your data, you do reduce your power because you're not estimating the size of the effect from both sexes simultaneously. And you don't ask whether it depends on sex, that interaction tool. And if you pool it, you can get data that looks like this diagram on the right. On the left, what I've done here is I've randomly sampled some data from a defined distribution, and I've added an effect that occurs in only one sex. When you pool the data, it just increases the variance, and it looks like your data is more variable, and you can't see the treatment effect. As soon as you disaggregate the data in line with the SAGA guidelines, you can start to see it depends on sex. And in this case, what would happen is the interaction term would become significant, and you would see, actually, I've got some very interesting biology that depends on the sex of the animals. So why is the factorial different? And it's because it's sort of pooling the data together. I don't seem to have a pointer, but let's go with this. So here, when you're estimating the effects of sex, you're actually pooling the control and treated for one sex and the control and treated for another sex, and you're estimating the average effect of sex. And likewise for treatment, you will be using the control position on the left. That would be the blue dot there on the left is showing you the average reading for the controls. You've got the blue dot for between the males and the females on the right, and that would be the average generalizable estimate of the effect of the intervention. When you're testing the interaction, you're comparing the differences, the slopes. And if the slopes have a similar size, then it won't be statistically significant. But if the slopes are very different, this suggests that there is an interaction effect, and the intervention depends on sex. And then you as a community have to make a decision for your science. Does that interaction, just because it's statistically significant doesn't mean it's biologically interesting. You have to decide based on the size of the effect whether I need to work with the average effect or whether I need to go, this is different depending on sex, this is biologically meaningful, I need to downstream work in a different way because it depends on the sex of the animals. So the interaction term is critical, and frequently people say to me, we will test the females once we understand the biology. Now, this is a flawed strategy, because if you test the females on a separate day, like in 4 months' time, and your effect size is different, we don't know if that's because you've run an independent experiment and it's just natural variation. or if it depended on sex. You have to do it at the same time. And in reality, I don't think you ever get to the second sex. I think that's just a placebo statement to make it feel okay. And it's also a structurally biased research approach. I would argue saying I'm going to study females later suggests that male biology is the truth, and you're going to work out how females differ. And that is, I would argue, is a flawed strategy, and it introduces challenges in representing women in science. I would argue a lot of the resistance is fear of change. I hear, to date, sex hasn't explained variation in my model. Well, one, it doesn't matter whether sex hasn't or hasn't. Including of both sexes isn't about finding sex differences. Including of both sexes is about having a generalizable estimate. So it's great news that sex didn't matter. That's a success story. It also could be that you haven't been analyzing your data well, and you don't know when that sex difference is going to occur. My prior work has only considered in one sex. And yes, that is a reality that many of us are facing. You know, it's the same within AstraZeneca. We are prioritizing new models where we're introducing new ways and models into the facilities and then to our research program. That's where we start trialing two sexes and demonstrating how it works and addressing the challenges of being inclusive. You have to, at some point, draw a line and make a change. And here I've got a sort of conversation I had with someone who's very, very senior in the topic. And I want to show that actually this is why I think it's often fear. My research, she said, I only ever study one sex. And I would go, well, why not both? And they would say, well, I can't because it would double the sample size, and that's not ethical. I said, you don't need to double the sample size, you just need to embrace factorial designs. And you'll estimate it across both sexes. And then they respond, I can't do that because the effect would be different between the two sexes. And I think she means a baseline difference. And I go, well, that's all right. If it's a baseline difference, the factorial design would take effect. And from Ben's paper, we know that provided the effect's in the same direction, you're just going to estimate an average effect. You only lose power to estimate your treatment effect if it's in the opposite direction or only in one sex, at which point the power passes to the interaction. I said, it'll be okay. She said, no, the effect is too different. I have to have an individual estimate. We just don't know, and it would be too risky to assume it would be the same. Well, if it's too risky it's the same, why are you only studying one sex? And this is someone who's actually really influential in the UK research scene. And to me, this is just our emotional reaction. It isn't that they don't believe sex doesn't matter, but it's our worries and anxieties that are playing out here. Now, this doesn't mean that I always think that you can study both sexes. There is still a need to make sure this is a viable experiment. But what we're looking for as a community is to move away from justification based on misconceptions and emotive reactions to a justification that considers logistical, ethical, or cost implications versus the benefits of including both animals in a research proposal. So in conclusion, I feel that research suggests that sex is a significant source of variation for both in vivo and in vitro, which aligns with our underlying belief system as biologists. Improving translation requires us to embrace variation. We talk about precision medicine. I would say sex is binary. It's an easy first step to improve generalizability. It is 50% of the population. And I know there are other sources of variation, age and ethnicity, but let's start with an easier one. But sex bias is culturally embedded in our research pipeline. But many of those barriers are misconceptions. And so I'm looking to challenge your misconceptions, and we've got a workshop on more detail on this tomorrow if you'd like to come. Including both sexes is not at odds with a reduction mindset. And the expectation is changing. We need to work out how to consider sex mindfully and effectively and when it is the right time to do it or not. And thank you very much for listening.
Thank you, Natasha. That was absolutely fantastic. I think that has really clarified the issues for the audience. Now, if anyone has any questions, please move to the standing microphones that are at the edge of the room. Martin, I think. Martin,
Yes.
I can see it is you.
Yes.
Please.
I'm at number 3. Hello. Very nice. I have 2 questions. One is, most academic animal in vivo studies work with an N of 6 or 8, which probably is badly underpowered even if you don't consider sex. If I ask those colleagues, well, do half and half, my concern becomes a little that in the new sex-based groups with 3 and 4, you'll get anything you like with a lot of false positives and false negatives based on chance alone. But that so I don't feel that comfortable with it because the factorial design, and I teach statistics as well, so and I know where you're coming from, but if your original sample sizes for certain budgetary constraints and so forth very low, I'm not sure the factorial design helps a lot.
Okay.
And that's my second part, and they really raised, so I want to raise that point. So what we recently did in a major program that we said from the beginning, we do half of all studies in males, half of all studies in females. We include common controls, and if that yields signals, we then take the next step. And I would like to have your opinion on that.
Okay, so let's focus on the first answer. So it's been known for a long time that factorial designs are more efficient, and yet it hasn't landed that we've switched to factorial designs. So Ben and I ran simulations to construct for a typical in vivo experiment. So I think the N was 5, so 5. 5 per group per sex. And we showed and explored it, and the paper's been published in PLOS Biology 2 weeks ago, that you are powered, it is reproducible, it is robust, and we did that to do empirical evidence to address the fears. We have actually taken it all the way down to, if you were doing an experiment with just 4 animals and you split it into 2 and 2, the same patterns of behavior held. So I would recommend that you read the paper, but I can assure you that it does hold. Yes, you're not powered to see a small difference in a sexual dimorphic effect between the 2 sexes, but if you base this in the context, if that research had only an N of 6, you are actually powered to see what I call a Cohen's d, a standardized effect size difference of what, about 1.8, maybe? That's my guess, I can't remember. But it's quite large.
That's large.
So you're saying, I'm only interested in a large effect. So to then sort of, the only time you wouldn't be able to see a sexually dimorphic effect is when it becomes quite a small difference. But you've already implicitly decided that you're only interested in large effects anyway. So have a read of the paper. I hope it will reassure you that it does still work, the factorial design, even with an N of 6, it's okay to split the N across the 2.
And the second part of this, what do you think about within a program doing some male-only, some female-only studies, and all having some shared controls and taking that?
I would want to sit down and discuss it more, but I think you're confounding batch differences with treatment effects, and how would you know the common controls? I think I'd have to draw it out with you, but I think I'd be worried that you wouldn't be able to say that the difference wasn't due to a period of time or a sex difference. And so I don't think it could work.
We've got lots of questions here, Martin. I think you need to meet with Natasha afterwards. Dr. Rathod over there.
Thank you very much. There's a great summary of the, I'm at number 4, of the views out there. So for a number of years, in clinical studies at least, so randomized trials, sex has always been incorporated and as a mandatory thing. Do you think we're heading towards that in the preclinical state? In a few years, we're not going to talk about this, it's going to be a compulsory thing to do, or is that going to be impossible?
I think it is going to become more compulsory, but there are situations where you can't include both sexes as default. There are welfare and ethical issues that could be at play. There could be issues of sampling if you're working in an ecology study, getting hold of both sexes. So I don't think it's going to be 100% coverage, but I do think we're going to be changing that the default is to. But what we need to is, I find it really interesting from outside. I see some people work with male animals who say they can't possibly work with females because they're difficult, and I see some people working with females who tell me that males fight all the time when we're talking about both mice. So I find it's like embedded cultural norms that are at play, and what we need to do is learn from each other. Well, how do you do it? How do you implement this to actually make it a doable problem?
Thank you.
I think we've got time for one more question. Sorry, and there was a question here first.
Hi, Iris Jaffe from Tufts in Boston. That was a great talk. So I have two really comments. One is that, you mentioned that the NIH was one of the first in the US to require addressing sex as a biological variable, and I sat on the NIH study section in the 4 years that followed that and chaired the NIH study section for the last two. And so it was my job to determine whether people had addressed sex as a biological variable. And what I found, at least in the first 4 years, was the ways it was addressed was either to say, we will look at both sexes and mix them together, but we won't look for sex differences. Or sometimes even, the Cre recombinase is on the Y chromosome, so we can only do our study in males, and therefore that was a justification, which to me does not address the spirit of the requirement. And so I think it's important as agencies want to put this into effect to move the needle, to really be clear about what it means to address sex as a biological variable and to actually do the factorial design so you could identify sex differences. And then the other comment I'll make is that in my own work we look for sex differences, so we use the factorial design. And what I've found frequently is that we see the same effect in males and females, but the mechanism is different when we go to drill down on that. So another common response was, we looked at males and females, the effect was the same, so now we'll study the mechanism in males and it will be the same in both sexes. So I also caution that even if the effect is the same, so a drug has an impact in males and females, it may actually be working in a different way, and that's important to understand.
Well, I think that's a really interesting observation and would be worth sharing out into the community if you are allowed to share your data in that way, because I think it's, highlighting these risks is a really important next step. One of the things we're also working on, which would be shared in the workshop tomorrow, is we're working on a decision framework for evaluating justifications. It's a 12-question set of framework, and it's to move people past these misconceptions and to an evaluative response. The framework gives a red, yellow, and green outcome. So red is your justification is not appropriate, it's referencing a misconception. Amber is there's a bit of a warning about your strategy, and green is your justification is appropriate, be it it's a single sex or a double sex. And so this is what I'm being a bit more nuanced, but to support funding bodies and public, ethical review boards in evaluating the justifications. And we hope to get that out this year.
Well, that was a plug for the workshop, so everybody come tomorrow to the workshop. Thank you very much, Natasha, that was fantastic. So the next speaker is George Kararigas, who is Professor of Physiology at the Faculty of Medicine at the University of Iceland. And George is going to tell us about why study of the effects of biological sex in cardiovascular physiology is important.
Okay, so Amrita, thank you so much. Thank you for this kind invitation and the opportunity to be here today with all of you. I have to admit I'm more excited about the discussion actually rather than presenting you some of our previous data. Natasha, thank you so much for setting the scene and also going through the different terminology that we're using in the field. I will also touch upon this a little bit. And it was a great question, Iris. I don't see you now in the audience. Actually, because Natasha was showing in one of her slides, in the exception slide, you were saying that there's no justification, or let's say one sex can be studied at the molecular level, but as Iris was saying, I mean, we do see a lot of times the phenotype being the same or comparable between the sexes, but the mechanisms, so the molecular mechanisms, they are different actually. So I think that even if we're looking at protein-protein interactions, I think there is a lot of value there to study both sexes. So my work is primarily focused on the cardiovascular disease, and I will be showing to you some of our previous data, and I will try to convince you why it is important to study the role of biological sex in cardiovascular physiology. Now, this is the World Congress of Pharmacology, and most likely you will know that the current standard medical care is the one drug, one dose fits all approach. Natasha was saying we have no pointer, so okay, maybe I can get the mouse.
No, the mouse works.
So,
the
yeah,
mouse works.
Yes, the mouse works. So with the one drug, one dose fits all approach, we end up primarily with 3 groups of patients. One group that will benefit, one group that will see no major effect, actually, neither positive nor negative, and one group that will develop adverse effects, actually. So just by, and again, just in inverted commas, as Natasha was also suggesting, by including sex and gender, together, of course, with the genetic makeup, we will go a step closer towards personalized medicine, which will basically lead to this situation, which will be patients benefiting through tailored therapy, either on the basis of dosing or even on the basis of changing the drug. So this is the cardiovascular track of the Congress, but focusing on the role of biological sex. And I think you will not be surprised that I can use the male and female symbols and still the unequal sign between them and still get away with this, because there are major sex differences in terms of prevalence. When we think of coronary heart disease, for example, you will appreciate that there is a higher proportion of male patients compared with the female patients. There are also important sex differences in terms of risk factors. I don't expect you to be able to read this, but let's say current or former smoking is more relevant as a risk factor for men, who are depicted here with a darker color. But if we think about hypertension, for example, it's a more relevant risk factor for women. There are also important sex differences in terms of presentation. Men usually present with typical symptoms, while women, they most frequently complain about some pain in the shoulder or some pain in the stomach area. Now, this leads to a major issue, and this is misdiagnosis. Female patients go to hospitals, they end up being misdiagnosed, they are sent home, and they eventually develop major effects, or they die even. So that's something to consider. We also have important sex differences in terms of pathophysiology. When we think about, again, coronary heart disease as a general term, men usually have localized stenosis, while women present with a more widespread thickening of the vessel wall. Now, for this situation, the male situation, we have guidelines for diagnosis and therapy, but for the female situation, we lack guidelines. There are also important sex differences in terms of therapy and treatment, and here sex and gender interact, but I will just stick to the sex for now. This is an old study from the Digitalis group. Back in 1997, if I remember correctly, their first paper in New England Journal of Medicine, not stratified by sex. They did not show any major effects, but a few years later, when they stratified their data by sex, they saw that the drug, and I'll be a little bit blunt, was killing the female patients. Last but not least, major sex differences in terms of prognosis or outcome. When we look at patients with heart failure, even if we separate them between heart failure with preserved ejection fraction and heart failure with reduced ejection fraction, there seems to be a higher proportion of male patients dying compared with the female patients. Of course, there are other things that we need to keep in mind, for example, diabetes, that they can make this picture just flip around. Now, just very briefly, because Natasha did a beautiful job setting the scene and going through the terminology, I have also included this in my presentation. When we talk about sex, we are considering biological aspects, while when we talk about gender, we are considering psychosocial and behavioral aspects. So if we look at this schematically, on this side of the slide, we are talking about biological sex. These are sex chromosomes and sex hormones. But when we look at this side of the slide, we're talking about gender. So we have stereotypes like once upon a time, the man used to be the breadwinner, for example. So both sex and gender, they interact together and they affect, together with other factors that I don't have time to discuss today, the development of disease and outcomes. The problem in the field is that many of these sex differences in pathophysiology are poorly understood. We know very little about the underlying mechanism, and there are hardly any sex-specific therapies. The solution seems to be pretty straightforward. We should try to characterize in detail these sex differences with the intention of elucidating the underlying mechanism and the ultimate goal of identifying novel sex-specific therapeutic targets. This, of course, needless to say, it would take us a step closer towards personalized healthcare. With this in mind, back in the lab, we're testing the overall hypothesis that biological sex and sex hormones, particularly estrogen and estrogen receptors, they regulate several biological processes and in response to stress-induced cardiovascular remodeling, and these include mitochondrial function, calcium handling, the development of fibrosis, and cell death. Our main aim is to characterize these sex differences in the context particularly of myocardial hypertrophy and heart failure, and to elucidate the underlying mechanism with the ultimate goal of identifying novel therapeutic targets. So a few years ago, what I did not mention is that my line of research is translational. So I move back and forth between humans and not just, again, in inverted commas, clinical data, but also clinical samples and animal models. So in this case, we started with patients with aortic valve stenosis undergoing AVR, so aortic valve replacement surgery. And we developed something that we call the maladaptive remodeling index. So this maladaptive remodeling index is underlain by an increase in left ventricular dimension, an increase in left ventricular mass, and a decrease in right wall thickness, relative wall thickness. We found that among the patients that we studied with aortic stenosis, there was a higher proportion of male patients presenting with this maladaptive index compared with the female patients. Of course, we tried to understand the underlying mechanisms. So what we did in this case, we went to the operating rooms and we collected small pieces of the hearts of those patients, so small biopsies from the ventricle or septum. And we performed genome-wide expression profiling. Of course, there was a large number of genes that was commonly regulated between the sexes. However, our surprise was the large number of genes that were either male-specific or female-specific. These are numbers. We try to understand biology behind numbers. So our pathway analysis showed that in the male patients, there were a number of pathways related to fibrosis. So extracellular matrix receptor interaction, for example, in TGF-beta signaling pathway that were upregulated, while in the female patients, we found that there were a number of different inflammation-related pathways that were downregulated. So we think definitely, although we might be looking at, again, similar phenotypes, the molecular mechanisms differ significantly between the sexes. So since this is an omic approach, we have to validate somehow our data, and I'm just showing you here some Western blot data for TGF-beta, as you can appreciate, with significantly higher levels in the male patients compared to the female patients. And I'm also showing you here staining for fibrosis. And as you can appreciate by the quantification of the data, there's a higher degree of fibrosis in the male aortic stenosis hearts compared to the female aortic stenosis hearts. So we concluded that in patients with aortic stenosis, so those patients that have pressure overload, there's a higher proportion of male patients with maladaptive remodeling, and this is underlain by a higher induction of profibrotic pathways, while the female patients seem to repress a number of inflammatory pathways. Now, more recently, because the biopsies that we used, they are multicellular tissues. So these effects that we saw, we don't really know which type of cells they were taking place. So we went back to the operating rooms. We recruited the same type of patients, and we collected again cardiac biopsies. But in this case, we isolated cardiac myocytes. And we took a targeted approach, so we moved from whole genome expression analysis to a PCR-based analysis. We focused on a number of genes that are involved in cardiac hypertrophy and remodeling. And as you can see here, all of those genes, the expression of all those genes was significantly higher in the male cardiomyocytes compared to the female cardiomyocytes. Now, yesterday, for those of you that might have been into the inflammasome session, you might have heard how important, or let's say how much we are understanding now about the importance of inflammation and the inflammatory component in cardiovascular disease, and particularly non-ischemic cardiovascular disease. So with this in our thoughts, we decided to focus on two inflammatory-related genes, NF-κB1 and CCN2, and we performed some kind of mathematical modeling, which showed that upon a twofold increase in the expression of those two genes, we identified a reduction in ejection fraction, which is an important functional parameter of the heart, in the male patients only. So we concluded at that time that there are pronounced sex differences in gene expression in isolated human cardiomyocytes. And we also found a significant male-specific association between cardiac function as assessed by ejection fraction and inflammation-related genes. So our next question was, okay, we cannot really manipulate our human patients. Can we use an animal model and mimic these sex differences? So we used the mouse and what we call the transverse aortic constriction, which is depicted here, to mimic pressure overload. And as you can see here, there is a development of myocardial hypertrophy at the end of the experiment. I know Iris had previously, in one of my previous talks, this question, so maybe I should address it already now. We have, maybe I should show first the data. So in response to pressure overload, we found that there was a significantly higher degree of hypertrophy in the male mice compared with the female mice. And when you have a stimulus, any type of stimulus, and you use both sexes, you need to think whether you are basically comparing apples with apples and not apples with pears, just to simplify things. So in many cases, using the same type of constriction, I'm just trying to simplify things a little bit, you might not have the same stimulus in both sexes. So in this case, we compared male and female mice being under the same conditions, just in a very simplified manner again. So we went back to our genome-wide expression profiling, and we were very pleased to see that this follows the same line as what we saw in our human patients. So significant increases in fibrosis-related genes like TGF-beta and collagens in the male mice compared to the female mice. And when we looked at fibrosis again, you will appreciate by the representative images here that there was a higher degree of fibrosis in the male TAC mice compared to the female TAC mice. So we concluded that the mouse TAC model is relevant for the study of sex differences in pressure overload-induced left ventricular hypertrophy. I'm speaking a little bit fast. I hope everybody can follow me, but I have a number of things I would like to share with you. So. Now, due to these sex differences and menopause-related differences that I have no time to discuss with you today, the overall hypothesis in the field is that the sex hormone 17-beta estradiol, or E2 in short, plays a major role along with its receptors, primarily estrogen receptor alpha and estrogen receptor beta. Now, most of you will think of estrogen as a female hormone, and of course it is mainly produced in the theca and granulosa cells of the ovaries. However, both men and women have estrogen, and this is through the local production of estrogen through the conversion of testosterone by aromatase. So both men and women produce estrogen, and there are several cases where actually the levels of estrogen in the circulation might be even significantly higher in men than in women. This occurs particularly in obesity because the adipose tissue is a major contributor of this local production of estrogen. Estrogen receptors have been reported in vascular endothelial cells, smooth muscle cells, cardiac fibroblasts, and myocytes. However, due to a number of, let's say, a little bit controversial data from particularly older trials, we still don't understand the effects of estrogen in the cardiovascular system very well. So there's a lot that we have to learn. So with this in mind, we went back to our mouse model, our TAC mouse model. In this case, we performed a proteomic analysis, and we used wild-type mice and estrogen receptor beta deleted mice. We call them BERCO. So these are mice that have estrogen receptor beta deleted. So as you can appreciate, I'm just showing you here the number of modulated protein spots, and I'm always comparing male TAC versus sham, and then female TAC versus sham for the wild type, and the same thing for the knockout mice. And as you can appreciate by the number of modulated protein spots, there was a higher number of proteins regulated in the male mice compared with the female mice. But upon deletion of estrogen receptor beta, there was a much higher number of proteins regulated in the female knockout mice. So again, for the sake of time, I'm not going into much detail here, but what we saw is that a number of proteins related to cytoskeleton and metabolism were decreased in wild-type male mice. At the same time, there was a number of proteins related to apoptosis and inflammation that their levels were increased in the knockout male mice. On the other hand, we saw primarily cytoskeletal proteins being maintained in the female mice. So again, this was an omic approach, so we need to kind of perform some validation, and I'm showing you here two important mitochondrial proteins whose levels basically were significantly decreased in male mice in response to TAC, while vinculin and cofilin, two cytoskeletal proteins, where their levels were significantly higher in pressure overload in female mice. So we concluded at the time that the estrogen receptor axis seems to be cardioprotective in females. So just for a few seconds here, just let's try this out. Let's see some hands. Excluding the feminizing effects of estrogen, who thinks that we should give E2 to males? Please raise your hands if you think that we should give E2 to males. Okay, we have one hand. Who thinks that we should not? Okay, wow. That's good. I'll show you the answer now with some data. So we went back to the clinic in this case, and we looked at patients with coronary heart disease and undergoing coronary artery bypass grafting. We collected, again, a little piece of their heart, and we developed an ex vivo model to be able to treat, basically, first of all, to keep these tissues alive in a dish and treat them with estrogen. And we performed our genome-wide expression profiling to understand the underlying pathways. So we were pleased to see that in response to estrogen, there was a large number of genes that were regulated in a sex-biased manner. One of those genes, in short, it's called MyLib. So things will get a little bit complicated here, so I will slow down a little bit. So in the male tissues treated with estrogen, we saw that the levels of MyLib were significantly higher compared with control. However, there was no effect in the female tissues. So we went back to our mice and we treated them in vivo with estrogen. We took, I should mention that we took middle-aged mice to try to avoid the female mice cycling, basically. And we extracted the hearts for isolation of cardiomyocytes, followed up with cell-shortening experiments or RNA protein measurements. Now, we were very pleased to see that exactly in the same way as we saw in the human cardiac tissues, in the hearts of male mice treated with estrogen, the levels of mylip were significantly increased compared with control, but there was no major effect in the hearts of female mice. Now, mylip stands for myosin regulatory light chain interacting protein. It's a long name. It's an E3 ubiquitin ligase, and its primary target, it's the myosin regulatory light chain, MRLC in short. MRLC is an important contractile protein. So we hypothesized that if we have too much of the bad guy, we would have too little of the good guy, and this is exactly what we saw. So in the hearts of male mice treated with estrogen, there was a significant reduction in the levels of MRLC compared with control, while there was no major effect in the female mice. So if we have too much of the bad guy, too little of the good guy, we would expect that function would also be affected. So the contractile capacity of those cardiomyocytes would be impaired. And this is exactly what we saw. Performing cell-shortening experiments, and I'm showing you here some representative pics between male control cardiomyocytes and estrogen-treated cardiomyocytes. And if we focus here on the quantification of the data, there was a significant decrease in the contractile capacity of cardiomyocytes coming from male estrogen-treated mice compared with control, while there was no significant, I should mention, effect in the female mice, if only there was a trend for a better, actually, function. Now, you might ask yourselves, why is this relevant for humans? The answer is coming from this study published in JAMA already a few years ago, where the authors looked at male patients with heart failure and different circulating E2 levels. And what they saw is that those male patients with heart failure and the highest E2 levels, or even the lowest E2 levels, they have the worst prognosis. So don't ask me what is happening here, but the data I just showed you, I think they contribute towards this picture. So the answer to my question is no. We should be very careful with giving E2 to males, since it seems that E2 exerts deleterious effects in male hearts and cardiomyocytes. So I hope that I have been within time. Just to summarize, I have shown you that in pressure overload, so primarily in patients with aortic stenosis, we found a higher proportion of male patients with maladaptive remodeling, which was underlain by a higher induction of profibrotic pathways. On the other hand, we found that there was an overall repression of inflammatory pathways in the female patients, something that we consider to be protective, actually. I also showed you pronounced sex differences in the gene expression of isolated human cardiomyocytes and a significant male-specific association between cardiac function and inflammation-related genes. The E2 estrogen receptor axis appears to be cardioprotective in female mice under pressure overload, and I showed you direct effects actually of estrogen on gene expression in the human myocardium that might be cardiomyocyte and sex-biased, basically, regulating cardiomyocyte contractile function. I would like to put forward that development of sex-specific therapies may contribute towards a more appropriate and personalized medical care. Now, if I was a little bit too fast and you've lost me, mostly, this is the most important slide and the take-home message. Now, there are important interactions between sex chromosomes and sex hormones, along with other factors that I had no time to discuss with you today that basically lead to sex and gender differences. And we need to take this into consideration if we want to move from this situation, which is the one drug, one dose fits all approach, so that we can come towards this situation, which will be a step closer to personalized healthcare. I recently moved from Berlin to Reykjavik at the University of Iceland, and I'm currently setting up my lab there. So I'm recruiting pre- and postdoctoral fellows. Iceland is a beautiful country. These are pictures that I have taken and the video that I have taken myself. So if you focus here for a few seconds, that's one of the geysers, the hot springs.
Oh.
Exactly, that's pretty amazing. Iceland might be an expensive country, but salaries are also good. So in case you're looking for a new position, please don't hesitate to contact me. Thank you very much for your attention.
Thank you very much, Professor Kararigas. We're now open for questions. Please come to the microphones. We've got just a few minutes, so short questions, please. But perhaps I might start. So I put my hand up saying, no, I didn't think you should give estrogen to male patients. But you sort of, I wonder what you think about testosterone. If you ask that question, how would you answer that? Is it exactly the same? Do you suspect that the androgen receptor pathways are negative in female sex?
It's not necessarily. Oh, I was thinking about testosterone in males. No, but you're saying about testosterone in females.
In women.
Yeah, in women. Yeah. I would think that in this case, if we think about, let's say, situations where there are higher androgen levels in women, like endogenous, I mean, there are a number of detrimental effects and a number of complications. So I would think that also exogenous administration would lead to a similar situation. So I would be cautious about that as well. But I thought that you were asking also, but maybe Astrid mentioned that, maybe there's a question. Yeah.
No, I just think that maybe when we're working on sex hormones, the focus at the moment is very much estrogen-led.
Very true.
But actually, possibly we should be looking at molecular pathways in cells of both sexes, but then looking at both of the key sex hormones too.
I just couldn't agree more with you. It's a pity and maybe I should say even unfortunate that we are, and I'm including myself, mostly focusing on estrogen and we're excluding androgens. I think this has to do primarily with the fact that we see a lot of protection in females. That's why we think that estrogen is a good guy and we try to understand what it's doing so that we can take it and also apply it to males somehow.
There's a question over there, I think.
Hello. Thanks so much for that fascinating talk. I had a few questions along the same lines, actually. One was, I think a lot of the confusion in the literature around cardiac remodeling and hormonal pathways in men and women seems to be around the way people are adjusting for also sex hormone binding globulins and the ratio of androgens to estrogens, and that this hasn't been standardized, and that people take incredibly different approaches, and it leads to quite a lot of divergent results. So I guess the first was a question in terms of how you envision the best way forward in managing, looking at sex hormone binding globulins and ratios of estrogens and androgens. And the second was in terms of limitations, actually, of looking at androgen levels in women and technology on big biobanks. So, for example, UK Biobank, the testosterone levels are with immunoassays, not with mass spec. So you end up with very low levels in women, and that limits what can be done, I think. So I guess those were my questions.
Yes, thank you. Yes, excellent questions, both of them. And this shows, just in a matter of a few seconds, how much we still need to do and we need to improve. Also on a practical, technical level. And I think that obviously there are a lot of issues in terms of when we think about gold standard techniques and mass spec, for example, to measure hormones. Can we, as Natasha was touching upon, also, you know, funding limitations as well? Can we go towards that? I would say that, of course, this is the obvious way forward. Whether this is practically doable or not, that's another thing. In terms of your first question, yes, there is a lot of controversy in the literature at the moment. There are a lot of mixed data, and I wouldn't have, let's say, a firm answer. Just I would say that we need to go into deeper detail and try to understand what are free circulating sex hormones doing, what are bound sex hormones doing, what is, let's say, the effect of the ratio between the two, if we think about, you know, androgens and estrogens. So there's still a lot we need to do.
So is it a short question?
I'll do it, I promise.
Very short, and
okay.
short answer.
Ulf Simonsen from Aarhus University, Denmark. And thank you very much for very nice studies. But I have one thing that keeps troubling me when I'm doing studies on female mice. It's because they are in diestrus. And you have shown also the mice which you have used, they were 2 months old, they're in diestrus. While the heart failure patients, they are certainly, most of them, postmenopausal. Which animal model should we use? And do you find the same if you take old female mice or young?
Yes, actually, I did mention that we used on purpose middle-aged mice, so they were between 14 and 16 months old, and the reason we chose these mice was to avoid the females from cycling, basically. There are two schools of thoughts. I have to be very short. Do they go through menopause or not, mice? What I can definitely say is that they stop cycling, and we have confirmed this with vaginal smears. It's pretty easy to do that, and that's just to avoid that.
Oh, that's interesting.
Yes.
Okay.
Thank you so much.
Well, thank you very much. Thank you. That was fantastic.
Okay, and it's with great pleasure I'd like to invite Ida up to the stage, who is from the University of Naples, and her poster was selected as it was the highest scoring poster, and she'll be talking about sex differences in B2-mediated response in mouth airways. Thank you very much.
Good morning. Before starting, I would like to thank the scientific committee for giving me this opportunity to share these results about sex differences in beta-2-mediated bronchorelaxation in mouse airways. It's widely reported that there is a significant sex difference in the airway physiology in lung function, and this is mainly due to the differences in chest structures, but also lung size between males and females. And besides this, sex hormones also modulate the immune response, which contributes to regulate the respiratory function. For instance, female hormones like estrogens are known for their pro-inflammatory role. On the contrary, androgens are known for their anti-inflammatory and immunosuppressive effect. And all this results in a sex difference in the incidence and severity of respiratory disease. And a well-known respiratory disease in which sex hormones modulate the incidence but also the phenotypes is asthma. Indeed, as we can see, asthma is different between males and females, but also changed at different stage of life. And during the adult age, when the estrogen's level is increased, women have higher prevalence of asthma, but also experience more severe symptoms. And an important consequence of this is the poor control of the disease in females. And this is partially due to the sex difference in the leukotrienes production, which is higher in female when the immune response is activated. Indeed, it has been proved that this sex difference in the leukotrienes production is due to the protective role of androgens, which is able to reduce the leukotriene signaling by modulating the 5-lipoxygenase enzymes localization. And in this context, we demonstrated that also in a murine model there is a sex difference in asthma, and as observed in humans, there is a sex difference in the leukotrienes production. As we can see, female following the allergen exposure exhibit a greater production of leukotrienes, and this is associated to an increase in the development of the asthma-like features when compared to males. And this sex difference is abrogated when female mice are treated with dihydrotestosterone, confirming the protective role of androgens in this context. And more interestingly, this sex difference is also associated to a sex-tailored response to anti-leukotrienes, as observed in clinical studies, but also in our previous paper. So based on these results, we hypothesized the possible sex difference in the asthma treatment, and we focused our attention on the beta-2 agonists that we know that are the most used drugs for the asthma treatment, thanks to their ability to give a quick relief from bronchoconstriction. And we know that these drugs are able to act directly on the airways muscles by activating the beta-2 receptor pathway and inducing the bronchorelaxation. But even if these are the most prescribed drugs for the asthma treatment, their efficacy is quite variable. Indeed, clinical studies confirm that factors like genetic polymorphisms, but also race or age, are able to modulate the beta-2 agonist response. So here we investigated the effect of sex hormones in the beta-2-mediated bronchorelaxation. And to do this, we used male and female biopsy mice that we have already demonstrated that show a sex difference in the T helper type 2 response, but also a sex difference in the airway reactivity. So we stimulated bronchi from males and females biopsy mice without any allergen exposure with the beta-2 agonist salbutamol. And we found that bronchi harvested from female are less responsive to the beta-2 agonist than bronchi harvested from males. So we wondered if this sex difference in the beta-2 agonist response was due to a sex difference in the beta-2 receptor expression. But Western blot, as well as the immunohistochemical analysis, confirmed that the expression of this receptor was the same between males and females. So at this point, we investigated the activation of this receptor, and we found an upregulation of the beta-arrestin-1 in lung harvested from female. This enzyme is involved in the internalization of the beta-2 receptor, and the direct consequence of the activation of this signaling is the reduced amount of cyclic AMP. So we measured the level of these mediators in bronchi from males and females, and as we can see, the upregulation of the beta-arrestin-1 is coupled to a slight reduction of the cyclic AMP level in bronchi from females, suggesting a possible sex difference in the activation of the beta-2 receptor. So next we went deeper in this signaling by evaluating the phosphodiesterase and pKa signaling. These enzymes are activated in a negative feedback when the cyclic AMP is overproduced, resulting in the degradation of this mediator. So we saw if there could be a sex difference in the expression of the phosphodiesterase 4A, which is the greater isoform in the bronchial tissue, and the Western blot confirmed the upregulation of these enzymes in bronchi from female. And interesting, these results was also reflected in functional studies. Indeed, when we inhibited the phosphodiesterase enzymes with IBMX, there was an increase in the beta-2 agonist response only in the bronchi harvested from female. And the same happened also when we inhibiting the pKa enzymes, which is responsible for the phosphodiesterase activation. Indeed, also in this case, we have an increase in the bronchial relaxation in females, but not in males mice. So these results suggested that the reduced response to salbutamol in female was due to an upregulation of the phosphodiesterase signaling, resulting in a reduced amount of cyclic AMP. So for this reason, we also evaluated the cyclic AMP level in bronchi from males and females, with or without the phosphodiesterase inhibition. And we can see that the inhibition of the phosphodiesterase enzymes increased the beta-2 agonist response in female, but also enhanced the cyclic AMP level in bronchi from females, but not from males, confirming the upregulation of the phosphodiesterase signaling in females. And these results was also confirmed in vivo. Indeed, the treatment of female mice with roflumilast, a selective inhibitor of the phosphodiesterase 4, was able to reduce the bronchial reactivity to carbachol, but more interesting was able to restore the beta-2 agonist response. So next, we investigated the direct effect of estradiol in this signaling, and we stimulated the bronchial epithelial cells from males with estradiol. And also in this case, there is an upregulation of the phosphodiesterase 4A expression. So at this point, we wondered if it could be possible to switch a male into a female response, and we did it by the in vivo hormonal modulation by treating male mice with estradiol every day for 7 days. And we used the same experimental protocol also to treat female with tamoxifen, a selective modulator of the estradiol receptor. And we observed that, as happened for the cellular studies, also the estradiol supplementation in male mice enhanced the phosphodiesterase 4A expression in the bronchial tissue. And this was coupled to the reduction of the beta-2-mediated bronchorelaxation in mice, as happened for the control females. On the contrary, when we modulated estradiol signaling in female, the beta-2 agonist response was significantly increased. So in conclusion, we can say that there is a sex difference in the beta-2-mediated bronchorelaxation, and in this context, estradiol signaling plays a key role by modulating the phosphodiesterase and pKa signaling. So all these support the idea that, of a sex-tailored treatment for the respiratory disease. I would like to thank all the people who take part in this project, and thank you for your kind attention.
Thank you very much, Ida, for that great talk, whirlwind through the topic. If anybody would like to give a talk, to ask a question, could they come forward to the mics? And in the meantime, I guess I had a question. One of the things I've often heard is people will say that sex differences won't translate. No, I got the impression that it did, that you felt there was a good translation between your mouse model and what was happening in the clinic.
Sorry, I didn't get it. Can you?
I've had pushback that there's no value in focusing on including both males and female because the sex differences that we see in clinic won't translate to a mouse model.
In this case, yes, I think that these could be translated in clinics, maybe for using the roflumilast, but I think that it could be better at the association with the beta-2 agonists only maybe for the female, for women, to enhance the response to the beta-2 agonists in the clinics.
Can I ask a question? So, I mean, you focused on PDE4 for entirely reasonable reasons. Do you think this is a characteristic of other PDEs? And should we be looking much further? I'm thinking actually about cyclic GMP levels in men and women and male and female mice. So you'd suspect that actually it's the other way for the relevant PDE in that case?
Yes, we focused maybe on the PDE4A because this one is the most expressed in the bronchial tissue, but also for the PDE5 that is involved in the GMP level, and there was a slight increase in the female, but there was a significant difference for the PDE4A. So we moved to this signaling, but maybe there could be a difference also for the PDE5.
Cool. Well, thank you very much for that great talk and welcome. I appreciate you joining us.
Thank you.
Okay, so our next speaker is Professor Pasi Tavi. It's just coming up the stairs. From the University of Eastern Finland. He's Professor of Cardiovascular Cell Physiology at, now let's see if I pronounce this correctly, at AI Virtanen Institute. Did I do that justice?
AI Virtanen.
Virtanen
Yeah, that's
Institute.
Hard
And Professor Tavi is going to talk to us about PGC1 alpha drives sex differences in heart failure susceptibility.
Thank you. Thanks for the invitation and thanks for introduction. Indeed, I'm going to tell you something about the PGC1 alpha and its potential role in determining some of the sex differences in the cardiac physiology and pathology. Since I'm a physiologist, I start by listing the physiological differences between male and female heart, some of which are pretty obvious. Females tend to have smaller hearts, partly because of the smaller body size, but female hearts are also weaker. And this data comes from studies with isolated cardiac preparations where it has been shown that the maximal force produced by the female tissue is actually less than in males. And it appears that some of these features in the contractility originates from single cardiomyocytes and the calcium signaling at the cardiomyocytes. And it has been shown that female cardiomyocytes have a smaller calcium transients and expression of some of the calcium handling proteins are different. There are also very well-known differences in the electrophysiology between males and females. Generally speaking, in most mammalian species, females have longer action potentials. And these electrophysiological differences arise from differential expression of certain potassium channels and some calcium channels. But I have been always interested in relationship between energy metabolism and the contractile function or the overall phenotype of the heart. Therefore, I will focus in this talk in talking about the energy metabolism. And to me, it's very interesting that it has been shown that in females, there are more mature and efficient mitochondria. And here it means that they are more efficient in producing ATP, and while doing so, they produce less ROS. They have consequently better ROS tolerance. But from the physiological point of view, I think that the most interesting thing in this energy metabolism is that the females have more adaptive energy metabolism. Whether they are facing endurance exercise or pathological load, they seem to cope with better. So when we talk about the energy metabolism, and especially transcriptional regulation of energy metabolism, it's hard to avoid talking of PGC1 alpha, which is considered to be the master regulator of energy metabolism. And it's by no means a new molecule. It was described 25 years ago. And then it was, soon after that, it was shown to be activated by endurance exercise, cold exposure, calorie restriction, or stress. And these stimuli activate a number of intermediates that regulate the expression or activity of PGC1 alpha. And when PGC1 alpha is activated, it binds to several transcription factors like PPARs, nuclear respiratory factors, and ERRs, and promote fatty acid import and oxidation, oxidative phosphorylation gene expression, and mitochondrial biogenesis. This still is true. This is the basic function of PGC1 alpha, but after 25 years of studies, we know much more of PGC1 alpha. We know that it's far more complicated than that. It has been reported that it has close to 10 splicing variants, all of which seems to have a different biological functions. and it's regulated by degradation. Its half-time is about from half hour to 2 hours, and it's targeted to number of post-translational modifications. It also has other binding partners in addition to those 3 that I mentioned, and these binding partners partly define its cell type specific effects. Importantly for these sessions and this topic is that expression of PGC1 alpha has been shown to be downregulated in a wide variety of different cardiovascular diseases. So as I mentioned, I'm interested to study the relationship between energy metabolism and the overall phenotype of the heart. And I saw PGC1 alpha as a switch. By turning it on and off, I can modify the energy metabolism at will and then study what happens to the overall phenotype of the heart. And so we did. This is from heart-specific PGC1 alpha knockout mouse model that we created. It's a typical myosin heavy chain driven Cree flox animals. And we first started analyzing the male animals at the age of 17 weeks. And yes, there was no surprise that the knockout produced progressive cardiomyopathy with a severe cardiac dilation leading to heart failure and death before 35 weeks of age. This heart failure was preceded by gradual decline of aerobic and anaerobic energy metabolism. And as shown in this mass spec data here, it was associated with the severe downregulation of many of the mitochondrial intermediates. And also, we saw that it reduces the number of mitochondria as well. So, then to the sex-specific issues. After seeing that, and because my student had collected data from both male and female animals, we asked the question that what happens, is there any sex specificity in this effect? And we started comparing male and female knockout animals. And yes, there was a difference, and to our surprise, the difference was so that the female animals tended to die close to 2 months before the males. And when we followed the cardiac function and compared the male and female knockouts, we could see that between the 12 and 16 weeks, ejection fraction went down and the ventricle volume went up at the same time when the ventricles got thinner. And this happened in the female knockout animals. Time point 2 weeks after that, we did echocardiography, in detailed echocardiography at the end point, and that confirmed the fact that in most of the structural and functional measures that we can get from the echocardiography, female knockout animals show signs of heart failure and cardiac dilation. Next obvious question was that we asked that, okay, is it due to the fact that there is some sort of a differential gene expression going on between males and females? So we did a bulk RNA sequencing, and the result was that the knockout changed the expression about the same amount of genes in both males and females. And in fact, males and females responded very similarly to the PGC alpha deficiency at the level of whole transcriptome and PGC1 alpha target genes. So that was a disappointment because this would have been an easy explanation for the findings. So we had to find other reasons for this female failure. So next, what we wanted to do is that, of course, to figure out how much the energy metabolism is actually suffering from the knockout. And we started to do isolated cells and studied cellular respiration by seahorse. And if we focus here in the maximal respiration and anaerobic metabolism, we can see that the male knockout animals show some decrease in the oxidation of glucose. But if we compare that to the female performance in the maximal respiration, female knockouts, there is downregulated anaerobic metabolism, oxidation of glucose, and fatty acids. Clearly showing that in females, the metabolic downregulation is more severe than males. This is all nice, but we don't know actually whether there is still sufficient energy for the females. So because of that, we started to study the AMP kinase, and especially AMP kinase phosphorylation. And female knockouts seem to have a slightly higher AMP kinase amount, but more importantly, there is much higher phosphorylation of AMP kinase. And AMP kinase phosphorylation is considered to be the marker of energy crisis, because that's one of the last things that the cells can do when they are facing energy crisis. So this indicates that at the metabolic level, the female cardiomyocytes are suffering from the knockout more than males. I mentioned about the electrophysiological differences between males and females, and we did some patch clamping to figure out whether there is any changes there. And to make a very long story shorter, we detected some baseline differences in the action potential length, L-type calcium current, and ultra-rapid potassium current, and all of them were equalized by the lack of PGC1 alpha. So these differences were not detected when we compared the knockouts together. Okay, then when we talk about heart failure and things like that, it's always possible that one of the reasons for the heart failure and contractile decline is at the level of individual cardiomyocytes and the calcium signaling in the cardiomyocytes. To study whether that is the case, we started to do confocal calcium imaging. We loaded the cells with fluor4 and did some confocal imaging. So this A figure shows the protocol we used that allowed us not only to record and characterize the basic features of the calcium transient that are triggered by excitation and that trigger the contraction, but also to evaluate the calcium fluxes through different calcium handling proteins like ranitidine leak, SERCA2, NCX, and plasma-lemmal calcium pump. So at the baseline, female myocytes have higher plasma-lemmal calcium pump activity and higher ranitidine receptor leak. And when we look at the knockouts, both male and female myocytes had smaller calcium transient amplitude. But if we then look at the difference, we can see that the female cardiomyocytes have really much lower calcium transient amplitude, even compared to the males. And this was accompanied with males and lower SERCA activity and the fractional calcium release. Fractional calcium release means that is a measure of how much from the releasable pool of calcium is released during each excitation. This is kind of not typical finding that you might expect to see when you analyzed failing cardiomyocytes. And especially so because it's not associated with the normal down on upregulation of certain calcium handling proteins that are usually associated with heart failure. So while there are changes in the calcium signal, we didn't believe that this is actually the whole truth. So what else can there be? If there are no changes in the calcium handling protein expression, could it be something else? And well, cardiomyocytes, they are actually giant cells, and yet they have to be able to contract very efficiently. And this requires that the calcium that triggers the contraction has to be released in all parts of the cell at the same time. And if this doesn't happen at the same time, then the calcium signaling is kind of, calcium transients are smaller and they last longer, and the contraction is compromised. So to figure out whether this is actually the reason for these things that we see, we started to do calcium imaging, again, confocal calcium imaging, line scanning, where a single line, laser line, is scanned at very high frequency over the whole width of the cell, and that creates a new image where each line represents one spot here in the cell. Intensity of the color indicates how much there is calcium, and the time goes from left to right. Sorry. And what we were actually looking is that assuming that there is loss of the synchronization of the calcium release, when we look at these local calcium releases, we should see a bigger variance between them, both in time and space. And this is actually what we did. And what we found is that when comparing all of these groups, the female knockout myocytes had higher variance in the spatial and temporal domain of the rise times and decays of these local small calcium signals. And then we did the same analysis analyzing longer time period to figure out if some of these spots that fail to release calcium, they are permanent kind of modifications or whether they come and go. And that indicated that indeed many of the reasons for this dyssynchrony is structural changes, or changes that last over beat-to-beat variation. What could it then be? To understand that, we have to understand the structure of cardiomyocytes, and there are specific structures that are intended to take care of the fact that the calcium is actually released in all places at the same time. And these structures are called T-tubuli. They are basically invaginations of plasma membrane, with the sole purpose of these is to guide the electrical excitation of the membrane inside the cell and couple the inflowing calcium to the rundown receptors and the SR and make the calcium happen in all parts of the cell at the same time. So as a final experiment, we started to label these T-tubular structures and in trying to analyze that whether there is something wrong with them. And yes, this is done at the latest phase, 22 weeks old animals, and we analyzed the density or the content of the T-tubuli, the areas that don't have T-tubuli, and then irregular T-tubular structures of different kinds. And in all these respects, both female and male cardiomyocytes showed some irregularity in the T-tubular structures, but clearly the females show a worse phenotype than males. I don't have any fancy molecular mechanisms to explain this, but the working hypothesis that I formulated is that maybe it is so that since the female knockout cardiomyocytes are suffering from the lack of energy and the T-tubular structures require constant maintenance, which requires energy, maybe the lack of energy is just compromising this maintenance and we see a T-tubular degradation. After seeing that and after Amrita asked me to give this talk, I was thinking that whether there's something else I could show you. And I remember that we analyzed, many years ago, we analyzed another PGC1 alpha mouse model, transgenic overexpression of PGC1 alpha that was originally described or done by Bruce Begelman's lab some 20 years ago, close. And they wanted to do, what they did is that they overexpressed PGC1 alpha using muscle creatine kinase promoter, and that produced a robust overexpression of PGC1 alpha in skeletal muscle and produced a unique phenotype in the skeletal muscle. But as a side effect, in this mouse model, there is about 1.5 to 3-fold increase in the PGC1 alpha in the heart. And it happens to be almost the same amount of PGC1 alpha overexpression that has been reported to occur after long-term endurance exercise. So I thought that this is a perfect model to study the role of PGC1 alpha in the exercise-induced changes in the heart. And this is, first we analyzed, or back then we analyzed male mice, and we did all sort of experiments, and we concluded that this transgene induces kind of athlete's heart type of phenotype in these animals. So there is increased cardiac volume together with the hypertrophy, among several other things. But when we also analyzed the gene expression by doing a bulk RNA sequencing, and we learned a lot about the PGC1 alpha induced expression by studying these animals. While PGC1 alpha is said to be the co-activator, of course, when you activate PGC1 alpha, you see that some genes go up, and there is also some downregulated genes. And we learned from here that in most cases, the downregulated genes impact the phenotype more than the upregulated. We also learned that PGC1 alpha regulates not only the genes involved in the energy metabolism, but also genes involved in contraction, growth, calcium signaling, and very heavily genes involved in circadian rhythm. Luckily, we had saved the data from these animals and some samples, so we started to do the bulk RNA sequencing to compare whether the transgene, whether there are differences between males and females in the transgenes. And to begin with, there seems to be, at the level of mRNA, there seems to be a similar amount of overexpression in both males and females. And according to the principal component analysis, the controls, males and females, are apart from each other. And when we look at the transgenes or the overexpression animals, they look to be closer to each other. And then comparing the gene changes, expressional changes here, between the controls, so males and females, there is a differential expression in 3,000 genes. But when we compare the transgenes together, there is only 50 genes difference, which is quite remarkable. And then the overexpression changes expression of 1,390 genes in males and close to 6,500 in females. This is a remarkable thing, and we are still analyzing why this happens and what else happens. But here this figure shows, or this picture shows what happens. That this is organized in a way that it's organized based on the genes that are changed in the overexpressing females in the metabolic genes. And if you look at the controls here, there is quite a big difference between the controls in these expression of these genes and the transgenic or the overexpression animals are much more like each other. And same happens if we do the same comparison concerning the genes that are involved in sarcolemmal functions or contraction or such. Okay, that work continues, and we are analyzing the echo data from these animals as we speak, but we'll see what comes out from there. To summarize the data that I showed you, it is clear that the lack of PGC1 alpha results in energy crisis and EC coupling defects, and it appears that the female mice were more sensitive to this deletion. At least they developed the heart failure earlier than the males. And also based on the overexpression study, it can be said that PGC1 alpha target gene expression is very sensitive to PGC1 alpha levels in females. And to combine the both findings, it is easy to conclude that low and high PGC1 alpha levels seems to equalize the sex-dependent differences in wide variety of different parameters. Of course, there is open question is that the biggest question is that what are the mechanisms determining the sex-specific effects of PGC1 alpha? Whether there is sex-specific binding partners or what, we are looking into it right now, but yeah, let's hope that we can figure it out at some point. This is the people from my group and our university, and then this whole PGC1 stuff was done together with Karolinska, George Ruas from Karolinska Institute. Thanks for listening.
Thank you very much, Professor Tavi. That was fascinating. In one of your, we're open for questions now. If you want to ask a question, please come up to the microphone. In one of your early slides, you showed that there are multiple pathways that influence PGC1 alpha levels. So are none of those changed at all between the sexes? Or different between the sexes?
Well, there are studies indicating that female hormones regulate PGC1 alpha. And definitely through that route, some of them are different to begin with between males and females. Back in the days, it must have been 10 years ago or so, we started testing whether we can actually change the PGC1 alpha expression or activity by using sex hormones. This was done in the cell models, and we couldn't detect anything. We used both the endogenous genes and the reporters, and we couldn't see anything. Maybe it's a context dependent or something like that, but definitely to answer your question, that yes, most definitely there are differences, but we didn't measure them all. Okay. This was more straightforward than that.
Okay, thank you. There's a question there.
Hi. That was excellent work. So in terms of the mechanism, we know that PGC1 alpha is a transcriptional coactivator for nuclear hormone receptors, and that includes the steroid hormone receptors. So I wonder if you've looked into whether the differences are related to PGC1 alpha acting as a coactivator for either the estrogen receptor or the androgen receptor. And in your bulk RNA sequencing data, are there differences in enrichment of estrogen receptor binding sites or androgen receptor binding sites in the genes that change when PGC1 alpha is knocked out?
Based on the expression data that we analyzed, we didn't see any sign of that. But however, I think that that's a valid question and should be studied further. As I said, that we are now studying, and one of the hypotheses that we have is that there has to be some sort of a sex-dependent or different sex-dependent partners for PGC1 alpha, and these, of course, would be one of those. But to make those experiments so in a more accessible model, that has been proven to be very, very difficult, because the cell models don't recapitulate the sex-dependent differences that well, and the whole animal is far too complex model to study these. But we are working on that, and that's definitely a relevant question.
There's a question over there on the right.
So thank you very much. I have a question regarding heart failure types. So is there anything known about the expression of the protein in heart failure with preserved ejection fraction, which is more a female-related disease, or where the incidence is higher in females, and heart failure with reduced ejection fraction?
Regarding the PGC1 alpha expression, unfortunately, I don't know. Maybe somebody else is better to answer that. But I went through the literature concerning the PGC1 alpha. If there are real differences between those conditions in PGC1 alpha, at least they haven't been reported that well.
Okay.
I couldn't find anything.
Thank you.
Okay. Thank you very much.
Thanks.
Thank you.
And I'd like to welcome to the stand our last speaker, Dr. Krishnaraj Rathod from the Queen Mary University, and he'll be speaking about the resolution of inflammation, women do it better, which sounds very interesting.
Thank you very much for the opportunity to present today. It's a great honor to be here. So I'm going to talk to you about resolution of inflammation and talk to you about some clinical work as well as translational work. So cardiovascular mortality has decreased over time over the last number of years, but as you can see in the last 5 years or so, the rates of deaths have plateaued a bit. We know that this is still a big issue. So globally, 620 million people suffer from cardiovascular disease. And this is about 1 in 3 deaths altogether, which is about 20.5 million deaths worldwide every year. So it's still a big issue. In terms of the rates of deaths in the UK, cancer is slightly higher, but in Europe, it seems to be the highest cause of mortality with almost 40%, which is higher than cancer and respiratory disease. If we break this down a little bit further, so all-cause mortality, the rates of mortality seem to increase over time, as you'd expect in terms of age, with the highest rate in the 85 plus. This is similar in cancer, so you have more mortality as you get older, which you'd expect as well. But cardiovascular disease is slightly different. What you see is in the younger age group, there is a higher rate of mortality in the males compared to the females, but something seems to happen around the age of 55 to 64, where female rates seem to increase again. And in fact, in the 75 to 84 group, the rates of mortality are very similar. And in fact, it's higher in the higher age group at 85. Some of this could be because women outlive men. So, but being female is not a good thing always. We know that women have double the risk of a second myocardial infarction after the first event compared to males, and this has been established a number of years ago. There's also higher rate of mortality after the first myocardial infarction in women, and again, this hasn't been studied that well, but we know that this happens. And women seem to have poorer outcomes after percutaneous coronary intervention. So that's a procedure that's carried out by cardiologists where we can open up blocked arteries after a heart attack, and we know women do a lot worse. And some of this is because of differences in the way that women are treated. So there's delays in treatment. We've already heard that women have different symptoms they present with. So they have pain in the epigastric area, in the shoulders, rather than your typical chest pain that men suffer. There's bias in the way we treat women. So women may come in with a heart attack, but they not go straight to the cath lab because the symptoms aren't clear or the ECG is not the same. And there's difference in the coronary anatomy. You heard that already. So women have diffuse disease, whereas men have more focal disease. Women have more tortuous disease as well. And there's differences in calcium burden as well, which means that the way they're treated is different. And again, looking at the sex hormone effect is very important because there are differences in that as well. And as I said already, women are not represented very well in clinical trials, and there's commissioning by the American Heart Association and the Lancet to include women in more clinical trials, because the majority of the data that we have probably applies to men and not to women, and that's where we're seeing the differences in the outcomes that we see already. So we decided to do a study under the mentorship of Professor Alwalia to look at differences between the sexes in terms of outcome after myocardial infarction, using a large data set. So we have a pan-London group, which consists of 8 hospitals, includes Bart's Heart Centre, Royal Free Hospital, the Royal Brompton, and the Harefield Hospital, which is now kind of merged in terms of the data we collect. St. George's Hospital in Southwest, Imperial College in the West, and in the South, King's College and Guy's and St. Thomas's. So this group was established in 2005, which consists of the 8 main centers that carry out percutaneous core intervention, or PCI. And what happened was in 2005, they all merged together and started to collect data in a kind of organized fashion. So I went around all the hospitals to collect this data in the year 2005, and now we have 125,000 patients in this registry. The data is collected as part of the part of the British Cardiovascular Interventional Society audit program, so it's of the highest standard, and it's maintained in a very high standard way. So what we did is we decided to look at this database and look at only the ST elevation myocardial infarction patients. These are the highest risk patients in terms of heart attack, and look at differences between the sexes and menopause and outcome after their procedure for PCI. So we compared men and women, and we subdivided the data further in the under the age of 55 group and the over the age of 55 group. Now the reason for choosing this is that the average age of menopause in the UK is 51, but there is a bit of a range. So between the age of 45 and 55, majority of the women undergo menopause. That's why we use this cutoff. Looking at the data now, so we had nearly 27,000 patients in this registry, which is one of the highest studied up to this point. But interestingly, just to concentrate on the call to balloon time, so these were significantly different in the sexes. So women had worse call to balloon time. So the call to balloon time is the time from when you make a phone call saying you've got chest pain to the paramedics, or 999 or 111, to the point where your arteries opened with a balloon or a wire in the cath lab. So this was much higher in the women compared to the men at all age groups, and even if we subdivided them in the under the age of 55 and over the age of 55, this was still present. Interestingly, the door to balloon time was very similar and not significantly different, which is a bit reassuring that once they get to the tertiary center where they have the procedure of PCI, there wasn't a difference between the groups, so we were treating these women and men equally at that point. But it seems that because of the differences in call to balloon time and the differences in possibly the symptomology, we were seeing some of the difference that I'm about to describe. So looking at the primary endpoint of the results, on the left is a landmark analysis. So there's a clear difference from the very beginning that women do a lot worse than men after their PCI procedure, and this goes out to 5 years. And there's a big difference. If we look at the subgroups, so in the under the age of 55 group, there was still a significant difference between the sexes. However, after correcting for some of the common known confounders using multivariate analysis, such as diabetes, hypertension, hypercholesterolemia, and smoking, and many others, we seem to lose this difference, suggesting that the women and men did not have a significant difference in the outcome after the procedure. Looking at the over the age of 55 group, again, there was a big difference, and after correction, we still maintained a difference in terms of worse outcome in the women compared to the men. So therefore, this suggests that there's a change in some biology in women, probably related to sex hormones, that suggests that women seem to do worse. To summarize this study, so the call to balloon times were a lot worse in women compared to the men, and this is still a big issue. We've not addressed this, even though primary PCI has been around for more than 30, nearly 40 years. We are saying increased risk of events post-PCI in women following PCI driven by events in postmenopausal women, so this needs to be addressed. There are differences persistent, irrespective of ethnicity and socioeconomic status, and due to time I wasn't able to show this, but in different ethnicities and different groups of social status, we are still saying the same worse outcome in women. So this has all been mentioned before in one of the talks, but we recommend a kind of precision medicine type approach where we can look at hormonal status at presentation to see if there's an indication that particular women may do worse following their infarct and procedure. So we therefore decided to also look at some mechanisms to work out why there are differences in women compared to men. And we developed 2 models of inflammation with the supervision of Professor Alwalia in her lab to look at this in a bit more detail. So we know that there's lower incidence of cardiovascular disease in premenopausal women, and the clinical study I just described the observational study went on to add to this evidence. And this differences is thought to be related to the female sex hormone. We know that cardiovascular disease is characterized by endothelial dysfunction, and systemic inflammation is a key driver in this process. But what we don't know is if there's differences between the sexes in terms of inflammation that drives this whole process. We therefore decided to use a low-grade systemic inflammatory model, so using the typhoid vaccine to look at differences between the sexes after a typhoid vaccine to look at endothelial dysfunction in healthy volunteers. We used a primary endpoint of FMD, or flow-mediated dilatation. This is a marker of endothelial dysfunction, and it was done using international standards that have been established already. I'll talk you through some of the data that's there already from before. So Professor Hingorani and colleagues in the year 2000 published in Circulation, where they gave a low-grade inflammatory systemic insult by giving the typhoid vaccine. And in these healthy volunteers, what they found was that 8 hours after the vaccine, there was up to a 25% reduction in their FMD. There was one person there that you can see has gone up with their FMD. And interestingly, there was one female in the study as well. And that will become quite important when I talk to you about our results. So this study was approved by the local ethics committee, and we carried out a similar study to what Hingorani and colleagues did. So on day 1, at pre-vaccine baseline, we would do some FMD measurements, take some blood samples to look at cytokines and chemokines and their systemic inflammatory response, and also samples for flow cytometry, as well as nitric oxide metabolites. On the second day, we would give them the vaccine in the morning, and then 8 hours after that, we would do the same measurements we did at baseline. The day after, this is 32 hours after the vaccine, we would do the same tests again, which would look at the resolution phase of the inflammation. That's our schematic for the study. We had 24 healthy volunteers in the study of each sex, so 12 men, 12 women. Some of the women were on the oral contraceptive pill, and that was reasonable, so they were still healthy. But all women were recruited at the same cycle in the menstrual period, which was at the mid-phase. It's 2 weeks into the cycle. So I'll talk you through how we measure the FMD, flow-mediated dilatation, if you're not familiar with that. So this, as I said, is a marker of endothelial function. What we do is use a ultrasound probe, put it in the brachial artery, and then we measure this for 1 minute, as shown in the top graph. We then inflate a cuff at suprasystolic levels at 300 millimeters mercury, which causes a reduction in the vessel diameter and causes your hand to go blue, causes ischemia essentially. After 5 minutes, we release the cuff, and this causes a hyperemia and essentially increase in the amount of blood to go to your hand again. And this is the reactive hyperemia occurs because of shear stress. So shear stress activates the endothelium and activates endothelial nitric oxide synthase producing nitric oxide, which causes the peak rise in the diameter of the vessel. So the FMD measurement is your peak response of the artery compared to your baseline. The artery then resolves back to the baseline, and we have our FMD response. Looking at the results now. So in terms of the baseline characteristics, the only main differences that we saw between the sexes was in terms of BMI and systolic blood pressure, and this has been described previously in other studies looking at men and women. To look at the systemic inflammatory response and to say if this was a truly systemic response and if it was low grade or not, we looked at the total white cell count. So the first bar in both sexes is pre-vaccine, colored in white. The red bar represents 8 hours post-vaccine, and you can see there's a significant increase in the total white cell counts, which suggests that there was a true insult. And this resolves very quickly back to the baseline at 32 hours, suggesting it's a low-grade temporary response. This was mainly due to neutrophilia. Looking at the primary endpoint now, so in the first graph for the males, again, the bars represent the same thing as I said before. So the white bars are pre-vaccine, red bars are 8 hours post-vaccine, and the green bar is a resolution. And we see a trend to a reduction in the FMD in the men. Now, this wasn't statistically significant, unlike the Hingorani study. However, there was still a trend. Interestingly, in the women, we were expecting the same thing, but what happened was there's a significant increase in their FMD response at 8 hours post-vaccine. So this was almost a doubling, which was then still present in the resolution phase, which is a very big surprise. And we were completely not expecting this. Now, the study was powered to see a statistical significant difference between the sexes in terms of a change in FMD from baseline to 8 hours post-vaccine. So what we found was that in the men, there was a 1% reduction in the FMD, but in the women, there was a 2.5% increase in the FMD at 8 hours post-vaccine. So the women seemed to have an improvement in the vascular function, whereas men had a reduction. Now, these changes could be completely independent of the endothelium. It could be all related to smooth muscle cell, because as you know, the smooth muscle cell can contract and cause vasodilatation and vasoconstriction. So we therefore decided to use a direct nitric oxide donor using glycerol trinitrate as under the tongue sublingually to see if there's differences. And in fact, we saw no significant differences in either sex over time, suggesting this is probably truly an endothelial phenomenon. So why are we seeing these differences between the sexes? Is it due to their systemic inflammation? Well, I already showed you that the data in terms of the rise in white cell count was very similar in both groups. So we therefore decided to look at the cellular mechanisms and to look at the reactivity state of the cells and the activation markers on cells. So in this, both of these group of patients, we used flow cytometry and looking at CD162 and CD62L, which are both involved in the recruitment of cells at the site of inflammation, we saw a significant reduction or dampening of the activation state of neutrophils in women in terms of CD162 and a trend towards a reduction in CD62L. We therefore decided to do another study to look at these mechanisms in a bit more detail, which I'll call the blister study, where we can have a window of opportunity to interrogate the actual mechanisms that are taking place. So the cantharidin or blister study was carried out by putting cantharidin on the skin. Now this is relatively difficult to do, particularly difficult to recruit because you have to have this blister to be placed on your participant. But what happens is that a very straightforward blister is formed on the skin, which is painless, which disappears after a few weeks and has no long-term side effects. And what you can do is by putting this blister, you accumulate fluid, which can then be aspirated, and you can look at leukocytes, cytokines, chemokines, and the activation markers I mentioned. So we concentrated on looking at CD162, CD62L, as they are involved in cell-to-cell interaction, and CD11B, which is involved in the transmigration of leukocytes to the site of inflammation. So we had 16 men and 16 women in the study. What we did is apply the blister on day 1 and day 3, and then aspirate the blister on day 4, which then gives us two types of blisters. So the first blister is a 72-hour blister, which looks at the resolution side of things, and the third blister is a 24-hour blister, looks at the acute inflammatory response. So it's a very nice model to look at this, and it's been already described and used in other studies previously. So what we found again is the baseline demographics were very similar between the two groups, apart from the differences in the BMI and the systolic blood pressure, as I said before. And the systemic inflammatory response in terms of total white cell count, the neutrophils, they were all very similar between the two groups, which then suggested this is truly a localized inflammatory model. So the interesting results now show that at the 24-hour blister, so all 16 participants in both groups had a blister that was there, none had resolved, and we could aspirate this for looking at the individual results. What was really exciting and interesting was that at the 72 hours, although in the men, none of the blisters had resolved, we had 4 that had popped because of technical issues. In the female, 9 blisters had resolved at the 72 hours, which is really interesting. Only 2 had popped because of technical issues, but we only had 5 blisters available for analysis at 72 hours, suggesting there's something happening in terms of the resolution. Looking at the total white cell count, so we had aspirated the blister, and there were 2 million cells on average over the time periods. There was a trend towards lower total cell count in the men, in the women at 24 and 72 hours, but not in the men. The total volume fluid was lower at 72 hours, as you'd expect, because there are only 5 blisters available for analysis. But what about this individual cell types? So looking at the neutrophils first, again, neutrophils had lower in terms of total number of cells at 72 hours and 24 hours, but this wasn't statistically significant. So this was a little bit disappointing. We were expecting differences in terms of the women, but neutrophils are the first cells that arrive at the site of inflammation, so you'd expect a high magnitude of response. So we therefore decided to look at the activation states of the cell, And looking at this, what we found was that CD162 and 62L was significantly lower at 72 hours in the females compared to the males, but not CD11B. So would this have an impact on the second type of cell that arrives at inflammation, the monocytes? Indeed it did. So in the monocytes, there was a significant lower number of total inflammatory monocytes in the females at 72 hours compared to the males. And again, looking at the cell subtypes, the activation state of these subtypes, we had a significantly lower CD162, 62 and 11B at 72 hours in the females compared to the males. So it's really, really kind of exciting. So what we're seeing is that we're seeing differences in the sexes in terms of the resolution side of things in terms of inflammation, not necessarily the acute inflammation aspect, and that seems to occur in both cases, which you need to essentially to survive. We therefore decided to look at some further cytokine and chemokine data to characterize this in a bit more detail. And we know that IL-8 is involved in the recruitment of neutrophils that drive resolution, and this was significantly higher in the females compared to the males. Other markers were CXCL1 and IL-10. So CXCL1 showed a trend towards higher levels in the females compared to the men. And IL-10 is an important marker because it's released by the M2 or non-classical monocytes that drive resolution. This showed a trend towards higher numbers in the females. Therefore, in summary, what we saw that the men did not have an improvement in FMB, in fact, had a trend towards worsening FMD compared to the women. Females seem to express improved FMD response post their vaccine. This was a truly new and exciting finding. And it's not a pro-inflammatory difference between the groups. So both men and women seem to have an appropriate inflammatory response, but it's to do with generalized reduction in the activation state of leukocytes in the females. So therefore, frequency of resolution at 72 hours was higher in the females compared to the men in terms of the blisters. Therefore, female sex protects against endothelial dysfunction induced by acute inflammation, probably due to accelerated resolution of inflammation. And to end, I'd like to thank everyone in our group at Barts and Queen Mary University London, but particularly to Professor Amrita Alvalia, who supervised me and mentored me over the last 20 years since I was a medical student to the point where I am now. Thank you very much.
Thank you very much for the very interesting talk, Krishnaraj Rathod. Is there any questions? Could you step forward to the mics? I guess I had one question from the beginning of the talk in particular, was then you were talking about the need to have a hormonal statement assessment before the intervention. Did you have enough data to look at whether HRT usage post-55 altered the outcome in the survival in that age group?
Yeah, it's a really good question. So unfortunately, this data set doesn't capture that data. It's a very cardiology-driven database, unfortunately. So at that time when it was set up, they didn't feel that it was an important factor. But there's more and more evidence suggesting that the previous fears of HRT may not be there in terms of the cancer risk, although there's still the worries about the thrombosis risk. And we're thinking about setting up studies and developing studies where we can incorporate HRT and sex hormones in ways that we can then have enough power to look at differences.
Well, thank you. And I think one person stepped forward there on the left.
Hi, I had a question about how the more rapid resolution of inflammation fits into what we know about sex differences. And the two situations that come to mind, one is pregnancy, which is a situation of relative immune suppression, where sex hormones suppress the immune system to prevent a response to fetal antigens that may be non-self. But then I also think of autoimmune disease, which is a lack of suppression of the immune system, which is more common in females. So I wonder if you could speculate on what might be the teleologic mechanism for all of this.
Yeah, and again, that's a really good question. So as you said, women have higher rates of systemic inflammatory diseases, and that's all immune related. It's difficult to say. I mean, I don't know if there's been a little bit of work looking at resolutional inflammation in arthritis, connective tissue disorders, and it could be that there's differences in the way that the sex hormones apply. So in pregnancy, there's a complete shift in how sex hormones work. There's differences in progesterone and estrogen to different levels that you expect from non-pregnant physiology, because pregnancy is a physiological phenomenon, and how, we don't know how this applies. So I think it's to do with, again, sex hormones, and the absolute levels probably make a huge impact.
Thank you.
Thank you. If there's no other questions, I really appreciate, we should give a congratulations to the talk.
Thank you very much.
Do you want to end the session?
So I think this has been a great session, and we've gone from experimental approaches to including sex as an experimental variable. We've been told off quite strongly by my co-chair, but we've also learned a little bit about some of the studies in the cardiovascular system that are showing us that it is very, very important to include sex in your experimental preclinical work. And then, of course, we're in this wonderful situation that we have all of these experimental models of cardiac disease that show sex differences that seem to simulate at least part of what we see in the human setting. And we have some potential ideas of what might be the differences between men and women. So hopefully this has been a session which is a good example of experimental studies in mice translating reasonably well to the human setting, and as long as we do our experiments correctly, hopefully we might have some novel pathways that we can use in therapeutics in the near future. Thank you for attending. Please remember that we have our workshop tomorrow, exploring some approaches to fixing the way that we do our preclinical experimental work, and we welcome you to that. Thank you very much for attending.